Welcome to the Salk Institute's Where Cures Begin podcast, where scientists talk about breakthrough discoveries with your hosts Ali Akmal and Brittany Fair.
Core facilities are resources shared by all Salk labs so that each lab doesn't have to develop its own expertise in, say, gene sequencing or specialized microscopy. Ken Diffenderfer directs a stem cell core facility, and we got a chance to talk with him about what exactly that involves. What does the stem cell core do?
Yeah, so our lab focuses on providing human stem cell models to researchers at the Institute for the purposes of studying aging, development, and disease and human cell systems.
Tell me a little bit about what stem cells are for people who don't know.
Yeah, absolutely. Great question. So stem cells are a type of cell that has an incredibly unique ability to turn into a different cell type.
And what do you mean by a different cell type?
Right. We are very complex multicellular organisms that cells are integrating together in organs, and organ systems are integrating to form us, right? All of that complexity comes from, ultimately a single cell, right? So the power of a stem cell to develop into all of these unique constitutive parts of organs, of organ systems, is really what makes them unique. This property that they have that allows them to do this is what we call differentiation. And so at the most basic level, a stem cell is a cell that can differentiate into something else.
Is that kind of like specializing?
Yeah, absolutely. You could think of it that way. Yeah. So the cell has, kind of no defined purpose, and then it makes life decisions. And over time, it becomes an incredibly specialized unit that has a very unique purpose inside of whatever part of the body it is potentially existing in.
That's super cool. You sound pretty excited when you talk about stem cells. How did you get into doing this?
Yeah. So my first exposure to stem cells was as an undergraduate at Cal State Channel Islands. And I had the opportunity to work with a lab there that was studying a neural stem cell line, and really using this as a kind of platform to see how some drugs that a chemist on campus was developing would kind of cause that cell line to react, right? Mm-hmm. Would it cause it to divide more, to divide less? Would it cause that neural stem cell to make specific types of neurons at higher frequency than others? But it really struck a nerve with me that these cells are incredibly powerful.
So you studied them as an undergrad, and then you went to grad school?
Yeah. So I went to graduate school also at Cal State Channel Islands. I did a master's program there, which had a stem cell technologies component to it. And a part of that was doing a year-long internship, which I did locally in the San Diego area at the Scripps Institute. That kind of led me into a position here at Salk.
And what are some of the exciting ways that stem cells can be used in research?
Oh, yeah. So, they are abundant and diverse. A few of the really exciting ways we use them here at the Salk Institute is through deriving patient-specific stem cell lines for the purpose of studying disease. And this is a technology that was developed by a Japanese scientist by the name of Shinya Yamanaka back in 2006, where we can effectively take a permanently differentiated cell, a cell that has made all of its life choices. It's no longer a stem cell. And we could access that material through a biopsy, through a blood draw, and take those cells and revert them all the way back to the early stages in human development.
How would you move that backwards in time?
Most oftentimes when we are doing this, the skin biopsy comes to us in what we call a punch biopsy. It's just a little kind of core of skin, roughly 3 millimeters in diameter. We'll take that little chunk of skin and we will treat it with enzymes overnight, and those enzymes are just breaking apart all of the individual cells that are kind of inside that biopsy. And then we can essentially plate those cells out onto a plastic dish, grow them out, expand them as they start to cover the entire surface of a dish, and then we can introduce 4 key genes to those cells. And these are the 4 key genes that Dr. Yamanaka discovered back in 2006. And so we use highly engineered viruses that have had all the disease-causing elements of them removed, and we essentially just use them as shuttles for genetic material. And so we will infect the cells with these viruses, and then allow them to make this transition from a permanently differentiated, a permanently developed cell all the way back to the earliest stages of human development, which we describe as pluripotency. And so that process takes roughly 3 to 4 weeks. And so it's essentially skin cells hanging out in a dish, and then over time they start to slightly change. And we see the skin cells change from these very elongated cells that cluster very tightly together into these very, very small, tightly compacted groups of cells that we call colonies. And these colonies are the induced pluripotent stem cells starting to form.
So at that point, those cells are capable of becoming a heart cell or a lung cell
Absolutely.
Or a brain cell?
At that point, those cells are pluripotent, which is the unique ability of a stem cell to turn into absolutely any cell type in the human body.
Wow.
And if we think about doing this in a patient-specific manner, like we can with the induced pluripotent stem cell technologies, most people get very, very excited. I have a relative who has Parkinson's. You can study that, right? I have a relative who has cystic fibrosis. You can study that. And so there is an immediate connection people can start to make to these technologies and see not only the value to kind of the academic community and the medical community as we kind of probe these diseases and better understand them, but also how these understandings can lead to better treatment, better therapies.
So help me get a picture of what the stem cell core looks like. Do you have big tubs of liquid that have undifferentiated stem cells just floating in them?
Yeah, that is a great question. We are literally right around the corner. Do you want to actually head over and take a tour?
You mean now?
Yeah, absolutely.
Sure.
I feel like we're on an episode of Radio Lab. Okay, so let's enter the core.
Okay.
If the door unlocks for us. There we go. Okay, Allie, well, welcome to the Salk Stem Cell Core.
Thanks, Ken.
We have a few specialized pieces of equipment that allow us to actually image the cell models that we work with. Some are just kind of standard microscopes that allow us to take a cell culture out of an incubator, put it on the stage of the microscope and visualize it. And some are actually a little bit more exciting. That's the one I want to show you right now.
The Yokogawa Cell Voyager 1000?
We have, yes, that. So this is a cool system that allows us to do very high resolution imaging, confocal style imaging. But this chamber is actually designed to keep the cells at 37 degrees Celsius, which is body temperature, and also injects a small amount of CO2 inside of them. And it mimics the exact same conditions that the cells grow in when we store them in the incubators.
Okay.
And so we can do imaging experiments over a very long course of time, program the software that runs the microscope to image every 5 minutes, an hour, over the course of a day, 2 weeks, a month.
Wow.
And really track, how cells are behaving over that time and monitor those changes visually. And this is the one we use most frequently. This is our IncuSite. So this one is actually a microscope that lives inside of an incubator. Okay. But what makes this one very cool is that it can actually image multiple plates at the exact same time. And it's actually imaging right now, so I can't open it and show you.
Kind of looks like a wine fridge, but for cells.
It does look like a wine fridge for cells, yeah. A little bit warmer than your wine fridge, yeah.
Okay.
Yeah, cool. So that's what we have in this bay. If we move down the hallway here, we can see another one of our bays. And this bay is a bay we have dedicated to media prep. So this is a little bit of a long corridor where we have some benches along one side, and then some shelving with various glassware that we use for production of media.
And when you say media, you're not talking about CNN or NPR.
No, I am not talking about those fantastic news outlets. I'm talking about the liquid that the cells grow in, right? And so this is our cell culture media. And a lot of the cell models we use here in the stem cell core have incredibly specialized criteria for that liquid media that they grow in. And so we have the luxury of having some expertise in what those requirements are here, and we can actually make these complicated medias in-house. And so we do a lot of that here. Okay. And this bay is not super busy now, but about every other week we do one of these very high-volume preps of this specialized media, and it's all done by hand. There's not robots or automation doing this. It is our small team of technicians and myself that are physically mixing these various components together and sterile filtering it and then providing it to our user base afterwards. Yeah.
So sort of like making custom cocktails.
Exactly. Yeah, but we don't drink this one, unfortunately. Yeah. Cool. All right, so moving down, if we turn to our right, we have one of our tissue culture bays. And these bays are absolutely the heart of the facility. This is where all the cell culture activity happens.
So what kind of cells do you have for us today?
Oh my gosh, we have so many things. The first thing I want to show you are some fibroblasts. Fibroblasts. So these are cells that we actually can derive from a small biopsy of human skin. These fibroblasts are one of the very, very common starting materials for doing that iPSC, or induced pluripotent stem cell derivation process, right? Okay. And the fibroblasts we're actually looking at today are from a collaboration with a large group of researchers at UCSD that study Alzheimer's disease. And so these fibroblasts are actually from an individual with Alzheimer's disease, and we are growing them out. And so this is the starting material right here. And before we actually get under the microscope, I just want to show you, if you look into the well here, and this is one of our weld plates. We have 24 little wells on this plate. And one of these wells has a very, very small amount of media in it, and it has a little disc of tissue that you might be able to see there. And that's actually the skin punch biopsy, just sitting on the very, very bottom of the plate. Wow. And what we do is when these biopsies come in, we treat them overnight with enzymes. And then the next day we'll come in, rinse off the enzymes, and then put that little small disc of tissue onto the bottom of the plate that's been coated with gelatin. The exact same gelatin you would use at home to make Jell-O with. It's just kind of a biological kind of cell culture grade of it. And then we let it attach to that gelatin and put the tiniest amount of media on it, just enough that it stays hydrated, but not so much that that media would go over the top of the tissue. Because if that happens, the tissue will start to float. And we want it to sit on that gelatin. And as it sits on that gelatin, little cells are going to start to crawl out of that biopsy. And that's actually how we derive the cells from this tissue.
Wow. Yeah, and you're controlling the microscope with the computer, is that?
Yeah, so this microscope does have a small little camera on it. And this really allows us all to look at the exact same thing at the exact same time, instead of everyone trying to squeeze onto the two little eyepieces. And so we have a small little camera that's just going to broadcast this image onto the computer screen here. Let me focus this in appropriately. So here's the edge of the tissue. Just this is a little small circular disc, and let's scan around it, see if we can see any cells. I think I see some. Get this focused appropriately. So if we look right at the edge here, it's a little bit hard to see on the screen. You can see we have these little cells, these little circular cells, right at the edge of this tissue, and they're kind of crawling out of the tissue there.
So the little translucent circles, is that what you mean?
Yeah, the ones attached to the bottom of the plate here.
Okay.
Oh yes, here's a much more dynamic area. So this is really a better example of that. And so you can see here, on the top part of the biopsy, this area is very active with cells that are crawling out of it.
To be clear, we're not seeing any motion in real time, but there is a greater concentration of cells at the edge of the biopsy tissue, whose movement outward would be obvious if we were to look at an image taken earlier in time. It almost looks like a reef in an ocean that you're seeing just below the surface of the water.
Yeah, absolutely. You could think of it like that. Yeah. So the next thing I'm going to show you guys are induced pluripotent stem cells. They're looking like a very, very compacted, tight group of cells that we would typically see in very early human development.
Like soon after a sperm has fertilized an egg?
Yeah, like 5 days after that.
Wow.
Yeah. But these cells are not from that process, right? These are induced pluripotent stem cells that have come from human skin that we've reprogrammed to the induced pluripotent stem cell state, but they still kind of mimic a lot of those qualities, a lot of those behaviors that we would see with an embryonic stem cell.
That's amazing.
So what I would show you next is one of the kind of exciting endpoints we can generate. And these are cardiomyocytes, or heart muscle cells.
Okay.
And the very, very cool thing about these cells is that as we go through the process of differentiating them from pluripotent stem cells, and they mature, the mature cells will do something very, very cool. Hopefully you guys can see these cells are actually rhythmically pulsating.
Yes.
And this is no trickery. I'm not shaking the plate. This is what these cells do as they mature.
That's crazy. They're all synced.
They are somewhat synced, right? Still not the dramatic organization of a 4-chamber heart that we would expect to see. But it's not a kind of random event, right? There is some organization, even just in a monolayer, single layer of cells.
Yeah, it's wild. At the earliest stages, they kind of know what they're meant to do.
Yep. So that's what we're looking at here.
That is really
beating
amazing.
Heart cells. Yeah.
So
it's hard
meditative.
To describe.
It's hard to stop looking.
It is very hard to stop looking. We get very geeky about this and love watching this when we have a bad day. We just come in and pull out the cardiomyocytes.
It's really calming.
Fawn over them. Yeah. We just need some like Zen type music in here. We can all get relaxed. Bust out the yoga mats.
Very cool.
Yeah.
And how long can these stay out like this?
Yeah, so the cardiomyocytes, in our hands anyways, are very sensitive to temperature change. So as these stay outside of the incubator, they're going to start to slow down. And eventually they'll stop beating. If we throw them back in the incubator, they'll start beating again within a few hours. Also, when we change media on these cells, we see the same thing happens. That they slow down or stop beating. And then if we throw them back in the incubator, within a few hours, they're beating again.
Okay.
So you guys actually, I rescued these for you when we were coming through earlier.
How's that?
One of our technicians here was about to change media on all of these. So I just caught him right before.
Oh, wow.
Otherwise, we probably wouldn't be looking at beating cells right now.
Wow.
Yeah.
They would have just been resting and getting used to the new media.
They would have been resting and getting used to the new media, exactly.
Oh, my gosh.
Yeah.
You've given us the grand tour hit all the highlights.
Thanks so much for coming by. I'm so glad we could share a little bit of the cells with you today and show you the facility.
Join us next time for more cutting-edge Salk science. At Salk, world-renowned scientists work together to explore big, bold ideas, from cancer to Alzheimer's, aging to climate change. Where Cures Begin is a production of the Salk Institute's Office of Communications. To learn more about the research discussed today, visit salk.edu/podcast.